Welcome to today's lesson on culturing microorganisms, specifically focusing on the vital process of preparing an uncontaminated sample using aseptic techniques. This is an essential skill in cell biology and pivotal for ensuring accurate and reliable results when studying bacteria. In our lesson today, we will explore the key aseptic techniques employed to culture bacteria without contamination, including sterilisation methods, safe handling practices, and the use of specific tools and media. These techniques are crucial for preparing uncontaminated bacterial cultures, which are necessary for precise scientific experiments. So far, you have learned the steps required to culture bacteria in the lab. This foundational understanding includes knowledge about the different growth media, incubation conditions, and basic lab procedures needed to support bacterial growth. Building on this foundation, today's lesson will uncover how these aseptic techniques are applied to advance your skills in microbiology. Now, let's dive into the core of today's lesson, the aseptic techniques used in culturing microorganisms. These techniques are fundamental in microbiology to ensure that our cultures remain uncontaminated, giving us accurate and reliable experimental data. First, let's look at sterilisation methods. Before use, Petri dishes and culture media must be sterilised. This can be done using an autoclave, which uses high-pressure steam to kill all forms of microorganisms. Alternatively, UV light can be used to sterilise surfaces by damaging the DNA of any potential contaminants. Inoculating loops are also important. These tools are used to transfer bacteria to the culture medium. They must be sterilised before each use by passing the loop through a flame. This process kills any lingering microbes that could contaminate the sample. Now, let's look at how we can ensure we handle our culture safely. Once the bacteria have been transferred to the growth medium, the lid of the Petri dish should be secured with tape. Importantly, the dish should not be sealed all the way around. This method helps to prevent airborne microorganisms from contaminating the culture, while still allowing for some air exchange to avert the growth of harmful anaerobic bacteria. After it has been sealed, the Petri dish should be placed upside down. This practice helps to minimize the risk of condensation drops from the lid falling onto the agar surface, which could cause the spread of bacteria that may contaminate the culture. Finally, let's take a look at the incubation conditions that are required when growing bacteria. Bacteria needs to be cultivated at specific temperatures, so we can use incubators to regulate this. In a school setting, bacterial cultures are typically incubated at 25 degrees Celsius. This temperature is low enough to reduce the risk of cultivating bacteria that could be harmful to humans, as many pathogenic bacteria thrive closer to human body temperature, which is 37 degrees Celsius. By following these aseptic techniques, we minimize the risk of contamination at every stage of the culturing process. This precision is essential for obtaining accurate and reliable results in any microbiological experiment, but also ensures our safety by preventing the accidental spread of harmful pathogens. Great work. That's the end of our lesson. Today, we focused on the importance of using aseptic techniques to prepare uncontaminated bacterial cultures. We discussed various key aseptic techniques, including sterilisation methods, such as using an autoclave or UV and sterilising inoculating loops. We also discussed safe handling practices, such as the proper storage of Petri dishes. Finally, we discussed incubation conditions for the cultures.
Watch the rest of the GCSE biology course at Sophos Education.